Hello everyone, this is part 2, so I'm going to be answering from question 5 to question 8. If you didn't watch part 2, make sure to watch part 1. This is for October 2024 for Unit 2 Biology, Pearson Edexcel. Let's get started. Okay, so question number 5. We have a type of bat that is endemic to Madagascar. So fruit bats transfer pollen from one baobab tree to another. The population of fruit bats in Madagascar is decreasing. The photograph shows one Grandidier's baobab tree in a field of rice and a Madagascan fruit bat. The species of tree is endangered. Identify 2 reasons why the species of trees is endangered. Use information above and the photograph to support your answer. Okay, so we have a bat and it's called a fruit bat, so therefore it eats fruit. Fruit produce flowers, therefore maybe this bat when it goes and eats the flowers, it pollinates something. This is what the images are trying to hint at you. We also have one tree which is endangered, and it's endemic to Madagascar. Endemic meaning it's only found in one specific geographical location, and this tree is around a rice field or a rice farm. Okay, so 2 reasons why this species of tree is endangered. Well, first of all, the cutting down of bamboo trees to grow rice, because they need to clear the land to make more agricultural land. And point 2 is that low pollination rate due to fewer fruit bats. Okay, maybe these trees produce fruit that these bats eat, and as they eat the fruit and they digest it, the seeds are spread in its feces. That could be another possible explanation. Okay, give 2 reasons how education could help conserve the Grandidier's baobab. So, number 1, we can teach people not to cut down the trees. We can promote the benefits of the trees. Number 2, we can promote the planting of saplings to increase the population. Okay, question B. Calcium ions are used to make molecules in plants. Explain the role of one named molecule that uses calcium ions. Okay, so the molecule is calcium pectate. What's the role of calcium pectate? Well, it holds the cell walls together. Now, water is also important to plants. Which of the following would transport water down the stem of a plant? Okay, so remember the xylem only transports in one direction, whereas the phloem is transverse in both directions. Therefore, the answer is going to be phloem and not xylem. Okay, the photograph shows pods and a soybean plant, and there's just an arrow to show you where the pods are. Okay, the effect of magnesium ions on the number of pods per soybean plant was investigated. In 2019, soybean plants were grown in 2 fields. One field had no magnesium added to the soil, and the other field had magnesium added to the soil. The mean number of pods per soybean plant was determined for both fields. In 2020, the investigation was repeated using the same fields. The graph shows the results of this investigation. Okay, so when I looked at this graph, number 1, I have no information stating that the soil in the fields was the same. And number 2, the magnesium ion concentration was not given. Okay, so give 2 conclusions that can be made using this data. As you can observe, in 2019, the mean number of pods per soybean is pretty low. Okay, so with no magnesium ions, which is the darker gray, you can see it's pretty low. It's maybe around 80, 87 or something like that. Whereas the one with magnesium ions, it's significantly greater. It's closer to 100. And in 2020, it's also the same trend. However, it's even a higher number of mean pods overall. So give 2 conclusions that can be made using this data. Number 1, there were more pods per soybean plant when magnesium ions were present. You can see that trend in both. The magnesium ions, which is the lighter gray, gave a higher mean number of pods. The second point is more pods per soybean plant in 2020 than in 2019. Okay, in another investigation, the effect of magnesium ions on the mass of soybean and maize grains was studied. The graph shows the results of this investigation. We have no magnesium and magnesium for soybean, and we also have the same for maize. And then we have on our y-axis the mass of 100 soybean or 100 maize grains per gram. So comment on the results from this investigation. Okay, so the first point is the presence of magnesium ions increased the mass of both plants. As you can see here on the diagram, so the presence of magnesium ions, which is over here and over here, this is higher than the one with no magnesium. Even though for the soybean, it's not that higher. Same thing for the maize. However, the error bars do not overlap, which indicates statistical significance. So my second point is going to be the mass of maize is greater than the mass of soybean. The third point is there is a significant statistical difference between maize and soybean due to no overlapping error bars. However, the concentration of magnesium ions was not given. You could also mention that the soil conditions or growth conditions were not given. Okay, question number 6. Okay, so in question number 6, in animal cells, the nucleus can divide by mitosis or meiosis. In the cell cycle, the time between two divisions of mitosis is called interphase. Describe what happens to DNA during interphase. Well, the first thing that's going to happen is that DNA unwraps from histones. Remember that chromosomes, the proteins, are wrapped tightly around histones. The second step is that DNA is checked for any errors. Therefore, the first point and the second point are going to grant you 2 marks. However, the third point, I just wanted to include it because transcription is then going to occur. Okay, question double I. Name the stage of mitosis that follows prophase. So remember our acronym PMAT. So it's prophase and then metaphase. Therefore, it's going to be metaphase. A person with Patau syndrome had 3 copies of chromosome 13 in each body cell. This occurred due to an error in meiosis and the formation of his father's sperm cells. The diagram shows what happened to chromosome 13 during the formation of the father's sperm cell. So we have chromosome 13 in the cell in prophase. Meiosis 1 occurs and everything looks normal. Now meiosis 2 occurs and sperm A and sperm B look perfectly normal. The centrosomes have separated. However, in sperm C it's empty and sperm D the chromosome moved down as if nothing happened. Okay, so a zygote is formed at fertilization. Explain how an error in meiosis could result in the production of a zygote with 3 copies of chromosome 13. Well, number 1 is the error happens when the centromere does not divide in meiosis 2. Just stating that it happens in meiosis 2 is going to grant you 1 mark. Therefore, the sister chromatids are not separated. As you can see in the previous diagram, the sister chromatids in sperm D were fully stuck together and not separated. Now, this resulted in some sperm cells containing 2 chromosomes of 13S. The zygote would develop into a morella and then into a blastocyst. Which row in the table is correct? Okay, so the zygote is obviously going to be totipotent. Why? Because totipotent cells are found in the morella stage in mammalian embryos. Okay, so the morella is a process in the development of the embryo. It's one of the stages. Okay, therefore the zygote is totipotent, which makes the cells in the blastocyst pluripotent. Now let's move on to question number C. An individual can also develop Patau syndrome if part of chromosome 13 becomes attached to a different chromosome during prophase 1. Suggest a reason why part of chromosome 13 can become attached to a different chromosome when a gamete is formed. Okay, so number one is that error is always, or not always, but sometimes error is occurring in crossing over. Okay, so during crossing overs, error usually occurs, or sometimes occurs, which leads to a mutation, or in this case, for this question, it's why the chromosome 13 was present in one sperm cell and not the other. Question number D. Okay, so methylation of DNA can alter the expression of certain genes on chromosome 13. Methylation of DNA can be passed on during mitotic cell division. Explain how DNA methylation can alter the expression of some gene. Okay, so methylation is the process of adding a methyl group to cytosine. So a methyl group is basically 1 carbon and 3 hydrogens. So a methyl group are added to cytosine. Then this will cause the genes to become deactivated or switched off. Explain why epigenetic changes to genes such as DNA methylation are passed on by mitosis. Okay, well, so DNA methylation is copied when the DNA is replicated, okay? Because when you have a gene that has had DNA methylation occur to it, when it's replicating when it's replicating, this methylation process does not go away. It does not go away. So therefore, the daughter cell will contain the same DNA methylation after the cell division. Question number 7. So cellulitis is an infection of the skin. It is caused when bacteria living on the skin surface enter through a cut in the skin. This results in swelling and redness of the infected area. The image shows a patient with cellulitis in one leg and one other type of skin bacteria that causes cellulitis. Okay, so this type of bacteria has a capsule, but not a flagellum. So this bacteria lives on the skin. It does not live in a liquid. So just one advantage of having a capsule to this type of bacteria. Okay, so remember that the capsule is sticky and moist. So one advantage is it's going to prevent being dehydrated. And the other advantage, which is the most important, is adhesion to the skin so that it can infect the person. Okay, question double I. Suggest why this type of bacteria does not need a flagellum. Okay, this question mentioned and told you that it lives on the skin. It does not live in a liquid environment. So it's going to need to have legs, if it lives on the skin. But it's not going to have a flagellum. But obviously it doesn't have legs. Okay, so just simply state, does not live in a liquid environment. Let's move on. Question triple I. Which of the following structures would be found in bacterial cell? Okay, so first of all, the cell membrane is present. The cell wall is present. Ribosomes are present. However, if they told you a cell wall made of cellulose, then it would be incorrect. However, just because it's cell wall, which is going to be made up of peptidoglycan, therefore our answer is going to be 1, 2, and 3, which is D. The antibiotic flucloxacillin is the current treatment for cellulitis. A student investigated the antimicrobial properties of said antibiotic. Let's call it antibiotic F. An agar plate had the type of bacteria which causes cellulitis spread over the surface of the agar. 3 discs containing the same concentration of antibiotic F were placed onto the plate as shown in the diagram. The plates were incubated for 24 hours at 25 degrees Celsius. We have the discs containing the antibiotic, which is the black dot, and the zone of inhibition where the bacteria had died. The table shows the students' results. We have the diameter of the zone of inhibition. We have 11, 7.5, 8.5, and the mean, which is 9. Okay, standard deviation can be calculated using this formula. The table shows some of the calculations done by the student. Okay, so let's find out what n is. So n is our sample size. Our sample size is 3 because x gave us a result for 11, 7.5, and 8.5. Okay, now let's find the mean. So the mean is going to be x with a line on top of it that represents the mean. So to find the mean, I'm simply going to add these numbers together and divide by 3, which gave me 9. Therefore, 9 is going to represent my x with a line on top of it. Okay, now let's fill in the gaps. So to fill this part, I did 7.5 minus 1.5. Sorry, I did 7.5 minus 9, which gave me minus 1.5. Okay, and then for this part over here, I simply looked up here and it said x, which is going to be 7.5 minus my mean to the power of 2. Okay, so my mean is 9, so it's going to be 7.5 minus 9 in brackets squared gave me 2.25. I put it in, and then I calculated the sum of these numbers. I added them together, which gave me 6.5. Okay, and then to calculate my standard deviation, I'm going to take the sum, which is 6.5 divided by 2, since 3 minus 1 is 2, which gave me 1.8. Okay, now question double I. Which of the following explains why the student would want to calculate the standard deviation for this experiment? Well, number 1 is to see how widely the diameter values differ from the mean diameter. That's what the standard deviation is. We want to see the deviation between values. So A is the question that makes most sense. Okay, question number C. Question C, not number C, that doesn't make sense. The effectiveness of treating cellulitis with antibiotic X, which is, let me try to pronounce that, flucloxacillin, alone was compared with a treatment that used both flucloxacillin and clindamycin. The diagram shows how two groups of patients with cellulitis were selected from an initial group of 2,444 patients. We have an initial group of 2,444 patients. Patients were not selected for this trial. Out of these initial patients, most of them were not allowed to continue. And then a selected group of 410 male patients with cellulitis on one leg were selected, 207 in group A, 203 in group B. What is the percentage of the initial group of patients that were placed in group B? Okay, that's pretty easy. Group B patient is 203, and since they mentioned initial group, the initial group is 2,444. So 203 divided by 2,444 times 100, which gives you 8.3%. So just how the study could be carried out as a double-blind test. Double-blind means that the doctor and the patient does not know who took what. So the patient or the doctor would know who took just flucloxacillin or both flucloxacillin and clindamycin. Now, in the mark scheme, you are not allowed to say who took the medicine and who did not. You need to specifically write down the names of the medicine. I don't know why. It's an annoying thing that you have to do, but it's just a lot of writing for 1 mark. But just make sure to mention the specific medications that they write by their name. Okay, the table shows the mean difference in circumference for the infected and non-infected legs at 5 and 10 days after treatment began. We have group A and group B after 5 days and 10 days. Comment on the results of this investigation. Okay, so the mean difference reduces over time. Group B had a greater overall reduction in swelling than Group B. The treatment for Group B is more effective. Okay, you can see why it's more effective because the mean zone of circumference between the infected leg and the infected leg has decreased. Now, only males is used in the study, so it cannot apply to females. That's one of the points you can also comment on this study. Okay, question number 8, which is the final question. A group of horses with the same specific characteristics is called a breed. The photograph shows a horse of the rare breed Akultiki that originated in Turkmenistan. The Akultiki population is in danger from inbreeding. Inbreeding means breeding horses that are related to each other. So there is a lack of genetic variation. A scientist suggested that the Akultiki breed is the same as an older breed known as the Turk Oman horse. Describe one piece of information that would need to be collected in order to determine if the scientists were correct. Okay, so first of all is DNA sequencing. So the information is DNA sequences. They can take saliva from the horses, or they can take blood, they can take multiple things. The reason is to see if the sequences from the breeds are similar. So DNA sequencing is going to produce a different bands of DNA, and they're just going to they're going to overlap them together to see what is similar and what is not. Okay. A scientist calculated the heterozygosity index of a population, 5,457 Akultiki horses, to be 0.661. How many of these horses are heterozygous? All you have to do is multiply the population by the index, which gave me 3,607. Okay, question C. The images show two rare breeds of horses, the Caspian horse and the Arabian horse. Caspian just means regions like Iran, Turkey, Armenia. These are the Caspian regions. And the Arabian horse, which is found in the Middle East in countries like Saudi Arabia, UAE, stuff like that. The Caspian breed was thought to be extinct until a small population was discovered in the mountainous regions in 1965. The Arabian breed is found in sandy deserts. The Caspian breed has small hooves in proportion to its mass, whereas the closely related Arabian breed has a large hoof in proportion to its mass. So just one reason for this difference. Okay, you can simply look at the images. One is standing on grass, one is standing on sand. Okay, so larger hoofs prevent Arabian horse from sinking into the sand. That's simply one easy mark. The table shows some information about one gene for a population of 400 Caspian horses. Okay, so complete the table to show how many horses would have the heterozygous genotype and how many would have the homozygous recessive genotype. Okay, so we have this equation, p squared plus 2pq plus q squared equals 1. Okay, I labeled what p squared represents, which is the homozygous dominant, 2pq heterozygous genotype, q squared homozygous recessive. Okay, so the first thing that you're going to want to do is you're going to want to find p squared, which is the homozygous dominant. So since we know that there are 46 individuals out of 400, we're going to divide to find the homozygous dominant, which is going to give you 0.339. Okay, therefore this is going to be our p, and we know the equation p plus q equals 1. We are going to rearrange that. Q is going to be 1 minus p. 1 minus p, which is 0.339, is going to give you 0.661 equals q. Then we're going to find q squared. Remember, q squared is for the homozygous recessive. And we're going to find 2pq for the heterozygous over here, and the two copies of the recessive alleles. Okay, now over here is the important part where people get confused. Once you find this number over here, the index number, you're going to have to multiply it by 400, or by the amount of population that you were given. Okay, because even when you're writing the exam, it's not going to make sense that you have only 0.4 horse has the heterozygous. No, that doesn't make sense. You need to multiply it by the population given to you to find how many horses have, and make sure that when you round it, it's a whole number. So when multiplying by 400, I just rounded it to a whole number, 179 and 175. Okay, breeding programs have been set up for both the Akultiki and Caspian breeds. Sperm samples have been taken from male horses from both the Akultiki and Caspian populations. Some of these male horses have since died. All of the sperm samples have been frozen. The effect of freezing and thawing sperm cells was investigated. The table shows the characteristics of two types of sperm cell samples. We have the type of sample, which is the fresh sperm cells, frozen and thawed sperm cells. We have the percentage of sperm that can swim, 50.7 and 26.9. There's a significant decrease. And sperm cell survival per hour, we have 117.7 and 48.5, which is a bit low. And then we have the percentage of sperm cells with damaged DNA. Now, as you can see, the percentage of frozen and thawed sperm cells has a much higher damage percentage. Okay, the graph shows the effect of the percentage of sperm cells that can swim on the number of successful pregnancies. Okay, we have the percentage of sperm that can swim and percentage of successful pregnancies. The correlation coefficient of the data shown in the graph is plus 0.9. So, discuss the advantage and disadvantage of using frozen sperm in breeding programs for these endangered breeds. Use all the information in question 8 to support your answer. Okay, so obviously, fresh sperm generally leads to better pregnancy outcomes due to higher motility, longer survival, and less DNA damage compared to frozen sperm. Studies show significant reductions in these factors after freezing. However, frozen sperm is invaluable for captive breeding. It allows collection for many males, captive and wild, facilitates breeding programs like artificial insemination to increase population numbers and prevent extinction. It eliminates the need to transport live males and enables genetic diversity management by selecting specific samples. So this prevents inbreeding. Frozen sperm sample also saves space and can be stored long term. The downside of frozen sperm includes a lower chance of successful fertilization and pregnancy due to reduced sperm quality. DNA damage from freezing can also lead to failed fertilization or unhealthy offspring. The process is costly, requiring specialized equipment and staff. Additionally, it's ineffective without viable females, and a significantly reduced pregnancy rate could negatively impact population recovery if not managed carefully. Nevertheless, a strong positive correlation exists between sperm motility and pregnancy success, highlighting the importance of sperm quality in both fresh and frozen samples. Thank you so much for listening to my video, and please subscribe and like and comment if this was helpful, or if it's not helpful, let me know what I can do better or what I cannot do.